Hello and welcome to the latest webinar, Enhanced Microbial Profiling and Metagenomics with Nanopore Sequencing. We have two fantastic presenters today, Dr. Chanwan Zhang and Dr. Stephen Batanovic, who will be sharing their valuable insights with us. Chanwan holds a postdoctoral position at the Chinese Academy of Agricultural Sciences with a focus on microbiome study using nanopore sequencing. Initially focusing on genomic and transcriptomic endeavours, he has delved deeply into the exploration of resource utilisation and functional regulation. Stephen completed his PhD at the University of Melbourne before pursuing postdoctoral research at La Trobe University and a research fellowship at Yokohama National University. His postdoctoral research employed molecular and genomic techniques, including nanopore sequencing, to investigate the interplay between bacteria and their natural predators. He has worked at Oxford Nanopore Technologies since 2023 as a field application scientist. Without further ado, I'll hand over to our first speaker, Dr. Zhang.
Hi everyone, thank you for staying with us. I'm excited to present our latest research on advancements in microbial community profiling using the newest Oxford Nanopore R10.4.1 flow cell. Currently, 16S ribosome RNA sequencing technologies, primarily including short-read sequencing, lead by the Illumina platform, followed by Microbial's full-length 16S rRNA sequencing. Illumina 16S is cost-effective and added a high number of sequencing reads, but has shorter read length. Microbial sequencing, on the other hand, offers long read length but is expensive, resulting in a low number of reads for analysis. Nanopore full-length 16S sequencing is based on the Oxford Nanopore sequencing platform, which enables acquisition of large amounts of full-length 16S rRNA sequences for microbiome diversity analysis. Nanopore sequencing is relatively novel and cost-effective. Why perform full-length 16S rRNA sequencing? Johnson reported that performing full-length sequencing of the V1 to V9 regions can achieve higher species resolution. In contrast, short-read sequencing can only target a specific region, resulting in a lower resolution, which generally allows for analysis only at the genus or phylum level. The Nanopore white paper also highlighted that nanopore sequencing has high specific level correlation and can identify more species. However, nanopore reads has a central error rate, but it can be improved through error correction. The average accuracy of the R9.4.1 flow cell is %92, but the corrected consistent sequencing accuracy can reach quality 40. Compared with the R9 flow cell, the R10 flow cell has become a dual recognition channel with the accuracy of up to quality 20. In the past, nanopore sequencing had a high error rate, and the research on nanopore 16S mainly focused on two aspects. The one was to improve accuracy through experimental methods, including INC-seq amplification or unique analysis identifier technologies, and to improve accuracy through multiple sequencing corrections, especially in chimeric proportion. On the other hand, improving algorithms such as nanoampli-seq. Many studies have been published to evaluate 16S rRNA data obtained through sequencing using R9 flow cell, but there has been no research on R10.4.1 data yet. Previous research have mainly focused on the following 5 points. For Minion sequencer R9 data, comparing the results of Nanopore and Illumina data. Focus on research at the level of phylum, family, and genus. Experimental method of improve analytical accuracy. Improving analysis accuracy through algorithms. In this study, we focus on 5 points. Evaluate the accuracy of quality 20 region in combination with R10.4.1 flow cell. error distribution and accuracy, performance at species and genus levels. What about annotations in different databases and different aligners? What's the difference between Microbial and Nanopore platform? In the current research work, using a custom workflow, NovaSeq, Microbial, Nanopore R9 and Nanopore R10.4.1 16S ribosomal RNA sequencing were performed on the Zymo and synthesis communities. To evaluate the taxonomic assignments, LAST and email for long-reads and data2 for NovaSeq classification tools were compared using the NCBI and Silva databases. For mock samples, we focus on the error profiles. The proportion of correctly classified reads, L1 distance, true positive taxa, false positive taxa, precision, recall, and F1 scores. For the environmental samples, we focus on the performances of R10 at the species and the comparison of Nanopore and Microbial, as well as correlations and difference from different diversity indices. We want to know how advantageous R10 has over R9. Then, can R10 reach a taxonomic assignment at the species level compared to Microbial? Globally, we showed that Nanopore R10 flow cells is capable and can be used for profiling the microbiota. This study used a commercial Zymo D6305 mock community, which contains 9 bacteria species with low 16S consistency. Also contains 2 fungi species. We selected 12 agricultural microorganisms to construct 2 synthesis agricultural communities. In these communities, species of the same genus but different species have a highly similar 16S sequence. These 2 synthetic agricultural communities, namely S1 and S2, using SYRB green qPCR technology to perform the genomic DNA copy number detection on 3 duplicates. The largest DNA value in the S1 sample was 1000 times the smallest value, and the mass value was dispersed in a gradient with a 943-fold difference in 16S copy number. In the S2 sample, a 20-fold difference in genomic DNA was maintained, with a 136-fold difference in 16S copy number. The Nanopore R10.4.1 platform obtained over 50,000 clean reads per sample. The average Q-score for raw data for the Nanopore platform was 19.77, while the data result primer it was 22.41. To evaluate the accuracy of the R10 flow cell, on the M-Plaken after improvements, error profiling was performed on the long-read data. We calculated the mismatch error rates and the indel error rates of samples. Meanwhile, obtained a mean error rate among all samples. We found that R10 flow cell greatly reduced the error rates, especially for insertion and deletion. We found that mismatch error rates reduced to less than half, insert error reduced to 1/3, deletion error rate reduced to least quarter. As the synthetic community has greater sequence diversity, the error rates of nanopore reads in the synthetic community are lower than those in Zymo. To demonstrate the existence of errors, we choose the sequence of diisofficins of different platforms with a single copy of the 16S gene. The IGV results show that R10 data performed well and had lower error rates. The similarity of 16S impacts the accuracy of species identification. At least 92.18% of the reads on the Microbial platform had a similarity of more than 99%. However, The similarity of the rates obtained via R9 platform is mostly between %92 and 95. Moreover, for R10 flow cell, more than %97 of identity was accounted for by ZIMO, %43.89, and by S1 %77.10, and by S2 67.26, respectively. Correctly assigned rates from the NCBI database were higher than those from the Silva database in the synthetic communities, but in ZIMO, an opposite trend was observed. While using the last NCBI method, the correctly assigned rates increase with increased accuracy. Interestingly, while using EMU NCBI at species level, R9 performs better than R10. Correctly assigned rates of the NCBI database were lower than those from the Silva database in the synthetic communities, but in ZIMO, an opposite trend was observed. In the synthetic community, the proportion of species and genus level annotations in the NCBI database is higher than that in the Silva database, but the opposite conclusion was observed in the ZIMO community. When comparing the NCBI databases, the annotation radio increases with the improvement of sequence accuracy. The annotation radio of Microbial data at the species level in the synthetic community is lower than its annotation radio in the ZIMO community. To quantify the results obtained from R10 in comparison to other platforms, the following metrics were used to evaluate the performance at both the genus and species levels. For the ZIMO simple, Microbial and Nanopore-based mapping method accurately detected the 8 true positive species. However, NovaSeq data was not suitable for species-level detection, which only detect 3 true positive species. For Microbial data, last Silva was used to obtain the latest false positive species with the lower L1 distance, while EMU NCBI processed the second lowest false positive species. For Nanopore data, R10 platform with email Silva produced the lowest false positive species. R9 platform was slightly better than R10 platform. When using the NCBI database across different methods, in conclusion, long reads get full recall. Microbial is better. Using last, the R10 platform gets a smaller L1 distance than the R9 platform across different databases. When using last classifier to compare the same database, the L1 distance as follows: Microbial less than R10 platform less than R9 platform. The L1 distance obtained using last Silva for Microbial data is the lowest. Illumina data has the maximum L1 distance. Microbial outperformed Nanopore for the ZIMO simple. The S1 simple was an intentionally challenging community containing several microbes of the same genus but different species with similar 16S sequences. At the species level, using the last NCBI method, the Nanopore data obtained the complete recall. However, one replicate of Microbial data detect 11 true positive species, not detect larval taller with an abundance of 0.0004. NovaSeq data can't achieve species-level identification. Software and databases contribute to the major difference in community abundance using long-read platforms. The NCBI database has fewer other categories than the email database. At the species level, the L1 distance of NCBI database is relatively small, but the genus level is equivalent. Microbial has the maximum L1 distance at the genus level. At different levels, the L1 distance generated by R10 data is lowest, such as email NCBI is 0.348, followed by R9 data, under the condition of obtaining a complete recall rate. R10 data obtained the lowest L1 distance, outperforming R9. Overall, Nanopore data performs best in the S1 community. Among the expected species in sample S2, all of the long-read platforms obtained complete recall at both the genus and species level by using last NCBI. Microbial gets far fewer false positives than Nanopore data. Then, email method may lack the detection of species of the same genus but different species. The performance of Microbial data is better than R10 data. The L1 distance obtained from the NCBI database is smaller than the distance obtained from the Silva database. L1 distance obtained by last NCBI method is Microbial less than R10 less than R9. So we think that NCBI database is more suitable for the complex communities in this study. We found that in the synthetic community, the Silva database has a bigger L1 distance and lower true positive. We have analyzed the specific possible reasons, which are mainly divided to three categories. Firstly, similar 16S RNA sequence lead to misclassification, such as the 16 sequence in the Silva database is highly similar to that of synthetic bacteria communities. In the Silva database, Azomonas was identified as Pseudomonas, but it didn't appear in the NCBI database. Another reason is different species in this genus have the same 16S sequence. Then synonyms and taxonomy rules and datasets also affect the results. In conclusion, the NCBI database is prepared for analysis of nanopore data at the species level. Mock communities can be used for evaluating the performance of different sequencing platforms, but they lack richness. Therefore, we analyzed the microbial compositions produced by different platforms using real samples, water, and soil. All samples were verified based on the minimal number of reads. As with mock, email successfully assigned all reads to species and genus level. Also, the rate of assigned reads by email was greater than that of last. The number of annotated species and genus was fewer. Use the LCA algorithm will lead to the judgment of poor quality data as unclassified. Furthermore, the number of species and genus identified in Nanopore R10 was higher than those identified via Microbial. However, the number of annotated species was more than that of Microbial. At the same time, R10 has more species than other methods, suggesting that sequencing errors don't lead to excessive identification of species and may be related to sequencing sensitivity or bias. Next, based on overall community composition, we compared the correlation between last and email class failure. For overall species composition results, we first compared the correlations between last and email aligners. The correlation between the two aligners was poor. In addition, we analyzed the average abundance of top 15 species across different platforms, aggregated them, and then calculated the correlations. For YW water samples, the average abundance of the top 15 species shared by Microbial and R10 has higher than that of Microbial and R9 at both species and genus levels. Comparing the number of top 15 common species in Microbial and R10, the last results was more accurate than email, and the correlation between last and email was not significant. Next, we focus on the results of last aligners. When the last aligners was used, the R10 and Microbial had a Pearson correlation coefficient of 0.51 for species level and 0.83 for genus level. Moreover, For NW water samples, the average abundance of the common top 15 species shared by Pacbial and R10 was no less than that of Pacbial and R9 at both species and genus levels. Pacbial and R10 have a correlation of 0.796 at species level and 0.911 at genus level, respectively. Furthermore, for HS cell samples, there were 10 common species and 9 genus detected by R10 and Pacbial, but only 6 common species and 9 genus were detected by R9 and Pacbial. The correlations at the species level was 0.99 and 0.985 at the genus level. These results suggest that the major species of R10 and Pacbial have a similar distribution, especially in HS cell samples. Box plot of alpha diversity across 3 platforms are shown. R10 data can obtain a higher Pacbial for CHO1 and future. In addition, Pacbial and R10 lie closer, Xiangnon and Simpson indicates, and their significance is lower than Pacbial and R9. Especially, Pacbial and R10 don't show statistical significance for YW water samples, but there were significant differences among Pacbial and R10. The results of rarefaction course showed that the immu aligner was easier to saturate because of the identification of few species, but using last aligners, R10 is often hard to saturate. For NW water samples, species saturation can be reached at 26,000 reads, but for HS and YW samples, more reads are required. Finally, we compared different platforms based on the pre-Curtis matrix. Current results suggest that species-level community composition was significantly diverse across different platforms and aligners based on Supernova and visualized by Pico for the unfiltered data. We found that the effect of aligners on the species composition is remarkable. In addition, we found that short-reads amplicon have more alpha diversity. We think that each species has multiple amplicon sequence variants, so the sequence diversity is rich. Finally, we calculate the sequencing cost. The cost of 50 nanopore sequences is nearly that of 30 Pacbial sequences, according to a recent promotion. The current study project per 10,000 reads is shown. Nanopore R10 is Chinese 50 yuan. Nanopore R9 is Chinese 45 yuan. Pacbial is Chinese 200 yuan. And NovaSeq is Chinese 30 yuan. Considering the advantages of high through output and the portability of nanopore sequencing, we believe that more researchers will employ nanopore sequencing to explore environmental microbes. Now, I would like to sum up the results of this investigation. We for the first time compared the Nanopore R10.4.1 amplicon with the Pacbial amplicon in 16S ribosomal RNA. In the synthetic community, percentage of 77.10 of the Nanopore R10 reads were more than %97 identity to the reference. Nanopore R10 achieved a good record at species levels. For environment samples, R10 can obtain more species than Pacbial. Pacbial and R10 are closer on the Xiangnon and Simpson index. Among the major species, R10 and Pacbial platforms reveal a similar microbiome. Both classifier and databases affect species composition. Overall, Nanopore R10.4.1 flow cell for the full-length 16S enabled species-level taxonomic identification for the environment samples. With thanks to the collaborators of this study, including Hanzhou Li and Jian Cao from Beijing and Xiaojin, and Zilin Ma, Hao Liao, Qiaoyun Huang, Wenli Chen from Huazhong Agricultural University. We offer a range of NGS Nanopore Microbial sequencing service for human, plant, animal, and microbial DNA and RNA analysis at highly competitive prices. We have established a sequencing experimental center in Hong Kong, which can receive samples from various places. Then, for Nanopore 16S data, many tools and online platforms have been released for data analysis, including Epitomy, NanoSPEC, BugSeq, NanoPep, and NanoOK, and so on. Finally, I introduce a new journal, iMeta. It publishes open access research that unites metagenomics and bioinformatics research. The new impact factor is 23. Thank you very much for the privilege of presenting this paper. If you are interested in my article, please communicate with me or cite it. Goodbye.
Brilliant. Thank you very much, Dr. Zhang, for that. We'll now want to move on to the next talk by Dr. Batanovic. Thank you very much for listening.
Thanks, Zhang, for your presentation. Hello, my name is Stephen Batinovic, and I'm a field application scientist at Oxford Nanopore Technologies. So today I'm going to present on how nanopore technology excels for use in microbial genomics applications. So Nanopore offers an all-in-one platform to approach microbial analysis. Today I'll focus on microbial isolate whole genome sequencing and de novo assembly, as well as full-length 16S sequencing, which can be used for high-resolution microbial community profiling. Of course, additional applications include targeted viral whole genome sequencing, such as that used for influenza and COVID sequencing, and then pathogen-agnostic metagenomics for pathogen detection or generation of MAGs. So what features of the Nanopore platform make it powerful in the implementation in these applications? So firstly, our instruments are highly accessible. So labs don't need to invest a great deal of cost just to obtain our sequencing devices. We actually provide the instruments for free in our starter pack model. We offer the MinION, which enables smaller size labs the ability to perform microbial genomics on-site or even in a field setting, as well as larger instruments such as the GridION, which is pictured on the left there, and our PromethION 2 devices for labs with higher throughput needs. Our library preparation protocols, such as the rapid barcoding kit, are streamlined, require minimal hands-on time, and are coupled with the ability to generate sequencing data in real time to deliver rapid turnaround from sample to answer. Lastly, the ability to generate long, accurate reads via our latest chemistry and base calling algorithms generate complete microbial genomes. So even prior to COVID-19, Nanopore established a key role in pathogen surveillance. Here we see two early high-profile efforts successfully using Nanopore for real-time monitoring of yellow fever virus and Ebola in South America and Africa, respectively. Of course, we then come to the global pandemic where Nanopore sequencing was heavily utilized for genomic surveillance of the COVID-19 infections through Arctic and midnight whole genome sequencing tiling methods. And these were used to rapidly and accurately surveil COVID outbreaks and virus evolution. Beyond the global pandemic, the community has now leaned into these approaches and are utilizing them for known and novel pathogens that relate to public health globally. So these include influenza and RSV sequencing. So here we can see several recent publications from the scientific community demonstrating how our latest sequencing chemistry is delivering high accuracy for a range of microbiology and infectious disease applications. These publications speak to the applications possible utilizing high-quality Q20 nanopore reads in both bacterial and viral de novo genome and metagenome assemblies, including sequence typing and also variant detection. So first up, I'm going to go over our new end-to-end Nanopore-only microbial isolate sequencing workflow, abbreviated to NOMIS. Genomics allows for the assembly of high-quality, de novo or reference-based microbial genomes with rapid turnaround time and very flexible sample batching. So one of the major benefits of nanopore sequencing is the ability to generate very long sequencing reads. So in comparison to short-read technologies that generate reads within 150 to 250 base pairs long, nanopore sequencing can give you reads that are tens of thousands of base pairs long. So this comes in particular handy when you're looking at resistance genes, for example, because these genes are often present on mobile genetic elements like plasmids or transposons. When we're characterizing bacteria in the context of outbreak investigations, we often don't only want to know what the resistant genes are, but where they're located in the genome and what else surrounds them. So when you sequence with short-read sequencing technologies, it's really difficult to get this information. Bacterial genomes have a lot of repetitive elements, and short sequencing reads are not often long enough to completely cover these regions. So even if you sequence the whole genome, if you try to stitch these reads back together, you'll end up with a bunch of short stretches of sequence, and you don't know how they fit together. So this is represented by the tangled assembly shown on the left. Whereas when you use long-read sequencing, these reads are able to span this repetitive region, actually able to recover the full circular chromosome and plasmid sequences. So instead of knowing exactly what the resistance gene is, you'll also know where in the genome that gene lies. Further to the advantage of long reads, nanopore sequencing also has the advantage of detecting any base modifications. So this is one of the upsides of the direct sensing platform that Nanopore uses, in that we can see the base modification data on every strand of native DNA. The challenge then becomes training our base calling algorithms to accurately detect the plethora of different modifications out there and the genomic contexts in which they occur. To this end, we've released new base calling models trained with native bacterial DNA, designed to fix any errors associated with methylated sites. And we now see that sequencing with our latest chemistry combined with these updated methylation-aware base calling models have improved bacterial genome reconstruction to enable reference quality assemblies without the need for short reads. And of course, with Nanopore, any base modification information is included for free. So this recent blog from Dr. Reinwick, based at the Doherty Institute here in Melbourne, Australia, discusses these new base calling models. So Dr. Wick's results demonstrated significant improvement in read accuracy with these new models, showing fewer errors compared to our previous model, and even greater improvements in consensus accuracy. And we can see on the right there that the consensus assembly accuracies reported for a panel of bacteria average out to about an assembly accuracy of Q60. So this roughly represents 1 error per megabase pair of genome. More excitingly, while these new models are now available for use within our Minnow software, our latest models released a few weeks ago offer further improvements around bacterial methylation motifs, leading to even further increases in these assembly accuracies. So these models are currently available within standalone data available on GitHub and will soon be available in our Minnow software. So what does our microbial sequencing workflow look like? So briefly, we can separate it out into 3 steps. So first is sample prep. So you can start from bacteria grown on solid agar or liquid culture, and then you move through 4 specifically designed cell lysis workflows based on the type of isolate you're trying to extract. So we have specific protocols for standard gram-positive and gram-negative bacteria, which use lysozyme or achromopeptidase for lysis assistance, as well as protocols for harder to lyse bacteria, such as Mycobacterium tuberculosis. We've also got protocols for fungi, and then just the universal bead beating protocol. So once you've extracted the DNA, you can then move into the library prep, and this uses our rapid chemistry featured in the rapid barcoding kit. So 200 nanograms of each purified DNA is mixed with a barcoded transposase mixture, which simultaneously cleaves and adds barcoded adapters onto your isolate DNA. You then pool your isolates, and then you subject this mixture to a rapid 5-minute incubation step where the sequencing adapters containing the motor protein required for sequencing are attached. The DNA library is then directly loaded onto a flow cell for sequencing for up to 72 hours. And like all nanopore sequencing experiments, the sequencing run once you've generated enough data. And then we move on to analysis. So our epidemic workflow for bacterial whole genome analysis is known as WF bacterial genomes. So epidemic can be run on any platform, Windows, Mac, or Linux, and via graphical or command line interfaces. So the workflow performs or reference-based assembly of bacterial genomes. If no reference is included, a de novo assembly will be completed using Fly, and then the draft assembly polished with Medaka. If a reference is provided, reads will be aligned to the reference, and variants will be called using Medaka. Of course, the workflow has a few optional extras. For example, it can run Proca to annotate any genes in your consensus assembly. And these results are then output into an intuitive report. So what else can the workflow do? So I've just mentioned that it can do generate high quality assemblies and do any gene annotation using Proca. But you can also do some additional analysis simply through checking additional checkboxes during the analysis setup. So these include MLST analysis, antimicrobial resistance profiling, as well as Salmonella serotyping. So here's an example of a bacterial isolate sequencing performed with our latest chemistry that was published earlier this year. So researchers from St. Paul's Hospital in Vancouver, Canada, performed whole genome sequencing on 56 multi-drug resistant Shigella isolates isolated from stool and blood between September 2021 and October 2022, and sequenced these isolates on a gridiron. So with Nanopore only sequence data, they were able to demonstrate near identical strain typing of the isolates using multiple MLST typing methods. They were also able to accurately assemble a conserved set of 10 plasmids within each isolate. Importantly, the whole genome sequencing antimicrobial gene detection was concordant with any phenotypic antimicrobial susceptibility testing, giving confidence that antibiotic susceptibility and resistance can be accurately predicted from whole genome sequencing data. So let's now change gears into a more targeted sequencing approach and see how Oxford Nanopore approaches microbial 16S sequencing. So as you're probably familiar, 16S sequencing is a predominant method for microbial identification in applications including microbiome characterization, food safety, clinical microbiology, and environmental monitoring. So current methods including Sanger and short-read sequencing provide limited sequence information, which often limits the ability to resolve reads to the species level. Conversely, the Nanopore method provides a strong value add to 16S profiling, as we have the ability to sequence and analyze the full-length 1.5 kilobase 16S amplicon, which has been shown to provide greater taxonomic resolution down to the species level. So here we see some information for our end-to-end 16S barcoding kit, which takes you through amplification library prep, sequencing, and analysis. So looking on the right there, first the microbial 16S DNA is amplified using specific 16S primers. So these are the 27 forward and 1492 reverse primers. After amplicon normalization and pooling, rapid attachment tags are then, which are present on those 16S primers, subsequently allow for rapid attachment of the nanopore sequencing adapters, including that all-important motor protein shown in purple. The library is then directly loaded onto a flow cell for sequencing up to 72 hours. And similar to the bacterial whole genome sequencing, the run can be stopped at any time once enough data has been generated. After sequencing, you can then perform downstream analysis using the Epitome 16S workflow, known as WF16S, to classify the amplicons in your sample. So the workflow has two modes of operation. It can either use Kraken2 or Minimap2 to determine the origin of your reads. So the Kraken2 mode is the default and faster method of the two, and employs a k-mer-based approach in the classification. Kraken2 can be used in real time, allowing the workflow to run continuously as your sequencing experiment is still ongoing. And the user can then visualize the classification of reads and species abundance in real time. The Minimap2 mode provides a finer resolution using an alignment-based method at the expense of more compute time. This method is ideal for any post-run analysis. Similar to the previous workflow, the 16S workflow provides intuitive reports and powerful graphics. And of course, a fantastic case study for the effectiveness of full-length 16S was just demonstrated by Zhang and colleagues. So I won't go into this too much, but just to summarize, Zhang's work aptly demonstrated the sequence quality improvements moving from our previous chemistry in POR to our newer generation chemistry in POR. And they were able to achieve raw amplicon read accuracy of 99%, which allowed them to deliver superior species level identification compared to short read methods. So thank you very much for listening.
Once again, thank you to our speakers and thank you to the audience for your participation. Thank you very much.